Last talk of this 20th ISMB. We have ISCB gives out two major awards every year. You have heard the Overton Prize winner. The nomination committee, the second award, is the award for the senior scientist. And the nomination committee, the awards committee, spends a lot of energy in choosing, very carefully choosing the candidate for the Senior Scientist Award, because it is, for us, the lighthouses or the stars in our field. It's a heavy competition, and it's a heavy fight in the committee to decide. I believe this year was a very clear decision, and I'm extremely happy to have Gunnar Von Heijne from Stockholm University win the prize this year as a senior scientist. Gunnar did his master in chemistry at the Royal Institute of Technology in Stockholm, did then his PhD in theoretical physics, but that already was some theoretical aspects of the structure, function, evolution of biological macromolecules, and in fact set the stage for his later work. He went on to become a reader in theoretical physics and then became a professor at Stockholm University in 1994. Remarkable in his career, he also was a science reporter in the radio from 82 to 85. And I believe this is a great thing to do other things, great way to do other things. 1997, he became member of the Royal Swedish Academy of Sciences. 2001 to 2009, he was the member of the Nobel Committee for Chemistry, which he also shared in 2007 to 2009. Gunnar has an ISI Hirsch index of 85, with 5 papers cited more than 2,000 times, 8 papers quoted more than 1,000 times, and 82 papers quoted more than 100 times. His most cited paper from 86 is a single author paper, New Method in NAR, Nucleic Acid Research, not Science, not Nature. Our field publishes the best papers in other journals. New method for predicting signal sequence cleavage sites. And that is the start to something that he really started. The study of the way proteins are signaled, proteins move around in cells, and the study of membrane proteins. When I came into this field a few years later, Gunnar was the one who got me into this problem of predicting membrane proteins. And he has been for many, many years the one who completely coined every technology, every idea, and every advance in the field. Later moved on to not only doing computational work, but also experimental work. An outstanding scientific track record. What I find even more remarkable, at the end of the day, ISMB is about people, as BJ correctly says. And Gunnar is an outstanding, what you'd say in New York, an outstanding mensch. He is incredibly shy. He's incredibly, when you come across him, you don't believe that this is the guy, the big man, the big scientist. And I believe this is very important as a role model. So for me, and also in that sense, and on the human level, Gunnar, I cannot thank you more for being the way you are. The prize has 2 aspects that we give. One is a lifetime membership. Gunnar is so young that leaves, in fact, we have $1,000 that I turned into a lifetime membership. And at his age, this still leaves a little bit of money for buying coffee. More precisely €500, and he decided to donate that to Travel Fellowship. Mind you, this is not money for his lab, this is money for his private pocket that goes to... So the first part of the prize that I want to award him now is the medal. Gunnar, congratulations. And the real thing, the second part of the prize, is the keynote. That's what we gain from him. Gunnar.
Well, thank you so much. I don't know if I'll be able to say hello to Burkhard after this without blushing every time. But, and also, of course, I was happy for the applause that I donated the $500. You should have seen BJ's email where she suggested that this might be a good idea. So thank you so much. This was okay. Right. So, thinking back, I've probably spent about 30 years of my life thinking about membrane proteins. And it's not easy to try to summarize or say something in a context like this about such a long time. But I don't think I'll feel too ashamed, especially since this is the 20th anniversary of the ISMB, to reminisce a bit. So there will be a little bit of reminiscing. But I'll also talk about stuff we're doing right now and the kind of problems that we're thinking about as we speak. So let me just briefly introduce membrane proteins, why we think membrane proteins are the greatest thing on Earth. Of course, membrane proteins are what mediates all contact between the cell and its surroundings. Everything, all the signaling, all the molecules that have to pass in and out of cells, all these transport processes are mediated by membrane proteins. So by that token alone, of course, they're extremely interesting from a biological point of view. Also, if you're interested in protein sequences, protein structures, and folding, membrane proteins have their special challenges because they do live in a very different environment compared to the water-soluble proteins that we're more familiar with. And in particular, as you can see on this nice MD rendering, that environment, the lipid bilayer, is a very, first of all, it's a very heterogeneous environment. It varies in chemical or physical chemistry over very short distances, like a few 10 angstroms. You go from from 55 molar water to essentially 0 molar water as you progress through the head groups. And so clearly, membrane proteins have to be designed in a very different way compared to soluble proteins. And this is a lot of what we've been thinking about for these 30 years is how is this special environment reflected in the way these proteins are designed? Can we apply, what kind of thinking do we need to apply to understand these, these structures, but also perhaps to predict certain aspects of these structures? So they're extremely interesting from a lot of points of view, and also they're very numerous. So estimates based on prediction methods tell us that something like a third of all genes encode membrane proteins, integral membrane proteins. This particular slide is a little exercise we did a year ago, taking some of the prediction programs that I try to identify membrane proteins and predict topology, and apply them to the human proteome. And what you can see is the number of predicted proteins with a number of predictors, and you see that they're fairly much in agreement, at least on the general aspects of this, of the proteome. And you see it plotted, the number of proteins plotted as versus the number of predicted transmembrane segments. So it's a lot of proteins with a single transmembrane span, and then the numbers decrease quite rapidly, except for one particular bump in the curve, which is 70 M proteins. And this, in higher eukaryotes, represents a very highly expanded gene family that codes for something called G protein-coupled receptors. So the bump at 70 Ms, it's basically all G protein-coupled receptors. Now, G protein-coupled receptors are a very interesting class of proteins, and they actually dominate the drug targets that the drug companies deal with at this point. So this is some sort of statistics on the targets for the main drugs that are sold across the world. Almost half of the drug targets are membrane proteins, despite the fact that it's 30% only that of the genes that code for membrane proteins, so they're disproportionately important. And you can see the big blue slice there, pi slice, is G protein coupled receptor. So, a third of all drugs are actually directed against these 800 or 900 proteins encoded in our genome and located mostly in the plasma membrane of our cells. So membrane proteins, have interesting biology. They have interesting protein structure-function relationships, and they are very important from a pharmacological point of view as well. So there are a good number of reasons why we're interested in those. But of course, like every field, the membrane protein field also has a long history, and you can, start it at different points. But from my point of view, and in particular because our main interest is really to understand the biogenesis of membrane proteins, the very first steps where these proteins get inserted into a target membrane. How does this work in the cell? And the very first idea, the very first sketchy idea of how this works was actually proposed back in 1971 by Gunnar Von Heijne and David Sabatini, who were not really thinking about membrane proteins at the time. They were thinking about secreted proteins. How are secreted proteins handled in the cell, and how do they get through a lipid bilayer in order to be secreted from the cell? And they came up with what's known as the signal hypothesis, basically postulating that a protein destined for secretion has a short signal peptide at the N-terminus, which is the address label. And that signal peptide will target the ribosome synthesizing that protein to some kind of export site on a particular membrane. In a mammalian cell, this would be the membrane of the endoplasmic reticulum, and in a bacterial cell, it would be the inner membrane. So they had this idea based on some fairly sketchy evidence that this is how protein secretion works, that you have a signal peptide targeting the ribosome to some kind of translocation site on the membrane, through which the polypeptide is then threaded as it's coming off the ribosome. So it's a co-translational process. Turns out they were basically right, and one kind of proof of that is that Gunnar Von Heijne was awarded the Nobel Prize in Medicine or Physiology some 20 years later. But from the point of view of membrane protein biogenesis, this was also a key proposal, because after all, a membrane protein is, you can view it as an incompletely secreted protein, a protein that has initiated translocation across the membrane, but that translocation process is then interrupted at some point, leaving the protein spanning the bilayer. And it turns out that precisely the same system that handles protein secretion also handles membrane protein assembly in both mammalian cells or eukaryotic cells and bacterial cells. So a lot of what we think about membrane protein assembly really goes back to these first insights back in the early 70s. Of course, in those days, nobody had a clear idea of what membrane proteins actually look like. There were no sequences around at the time. And the very first sequences came at about the mid-70s. So this is a paper from 1975, where by a lot of laborious protein sequencing, the sequence of a red blood cell surface protein called glycophorin A was pieced together. And the most interesting aspect of it is what's in blue, encircled in blue, and that is a stretch of about 20 hydrophobic amino acids. And it didn't take a lot of imagination to come up with the idea that this is what spans the membrane. It's these 20 hydrophobic amino acids that penetrate through the lipid bilayer. And if you think a little bit further, it's also pretty obvious that if you put a piece of hydrophobic polypeptide through a lipid bilayer, it's going to fold almost by necessity, has to fold into an alpha helix. That's going to be the most stable state of the membrane penetrating part. So from here, the idea of a hydrophobic transmembrane alpha helix was kind of has its birth at about this time. Actually, 1975 was a good year for membrane proteins because that's also when the first three-dimensional structure information came out. This is a protein called bacteriorhodopsin, and the structure, albeit at very low resolution, was solved by electron crystallography, not three-dimensional X-ray crystallography, but two-dimensional electron crystallography. And what became obvious immediately when you see the structure is that it's really built from 7 sausage-like densities that pack together into a bundle. So each of these sausages is obviously an alpha helix, and so this is a 70M transmembrane protein. So already in the mid-70s, it was, that's actually 75 is the year I started my PhD work. There were some early pieces of information about membrane proteins. So when I came into my PhD, which I did in a theoretical physics department in a group that I just started working on sort of theoretical biology problems, I got interested in protein sequence structure relationships, not initially membrane proteins, but more generally. And it's just fun to think back a bit, those were the days I spent my first year punching, at an IBM card puncher, basically punching protein sequences onto punch cards that we would then run in the computer. And I was very happy about a year or so later when we got our first 300 baud phone modem. So I didn't have to go down to the computer center every time I needed to put in a new protein sequence. So this was all nice and well, but of course what really changed the game was this. So after I got my first Apple II, I didn't have to worry about anything after that. So the early bioinformatics in the membrane protein field started about 1980, I would say. And that's about the time I also got interested in membrane proteins in particular. So one of the first things that happened was that people started to do hydrophobicity plots, scanning the amino acid sequence data and looking for hydrophobic segments that would be transmembrane helices. And of course, Kite and Doolittle is the most famous paper from that period. I think part of its fame is that they had a brilliant idea to put in the supplementary the actual code, which was probably about 25 lines or something of BASIC, that produced the hydrophobicity plot. So everybody could immediately punch it into their own computer and run these hydrophobicity plots. That was a brilliant idea of spreading the software, as it were, but in a little printed appendix. And a few years later, we were all very happy with the hydrophobic helices, but I started looking outside the membrane, and I started to look at the loops that connect the helices. And it was very quickly, it was obvious that the outside loops, those that face the exterior side of the cell, and the inside-facing loops in these multi-spanning hydrophobic proteins had a very different amino acid composition. And so what you see here is that the inside-facing loops, this is from about 20 proteins for which there was some sort of topology model at the time. The inside-facing loops have many more positively charged residues, lysines and arginines, than the outward-facing loops. And these two very simple concepts, hydrophobic helices and what later became known as the positive inside rule, these two very simple concepts actually turned out, in hindsight, to be extremely fruitful and really is still guiding a lot of the thinking when it comes to how membrane proteins insert into the membrane. And I'll just show you what this looks like in a real protein. So here it's just one membrane protein where the hydrophobic helices are noted in red. All the positively charged residues, lysines and arginines, are in blue, and you see in this particular case, for sure, they all concentrate on one side of the membrane. And you also have some tyrosines and tryptophans that were later found to kind of often flank these hydrophobic helices. So there are some fairly simple architectural principles that underlie this vast universe of membrane proteins, helical membrane proteins. Now, these early studies, hydrophobicity analysis, the positive inside rule, allowed people to start making predictors for the actual topology of membrane proteins. How many transmembrane helices, what's the orientation of the protein, just from sequence data. And we took part in some of this work, so some early topology predictors like TopRed, And after a few years, we teamed up with Søren Brunak's group in Copenhagen, and this has over the years led to a number of predictors, not only for topology like TMHMM with Erik Sonhammer and Anders Krug, but also for signal peptide predictors like signal P, and for more general localization predictors like target P. So this turned out to be a good area to apply bioinformatics techniques as well. Now, if I fast forward to more recent times, it's been fun to work in the membrane protein area this last decade or so, because it's really become a hype area. And these days, membrane proteins are, all in rage. And you can ask, why the hype? Why have membrane proteins become so much in the headlines in the past 10 years or so? And I think the simple answer is that it's only in the past 10 or 15 years that we have started to get enough structures of integral membrane proteins to actually understand something more detailed about the structure and function of membrane proteins. This is Steve White's famous plot showing, it's not quite an exponential increase, it's a little bit sub-exponential, but it's still a rapid increase in the number of structures. But it only started about, let's say, 10 or 15 years ago. That's really when it took off. And these days we're getting, quite a number of new structures every year. So this has really created a lot of excitement in the field, because there are a lot of interesting functions associated to these structures. And of course, it also immediately poses the problem of structure prediction. How much can we predict about structure given the information we have from other structures and from sequences and so on? So that's clearly a field that's ripe, I think, for progress. And just last week or a couple of weeks ago, there were two very promising papers, one in PNAS and one in Cell, showing that indeed structure prediction, three-dimensional structure prediction for membrane proteins seems to be on the horizon. So we kind of shifted our attention a little bit away from the bioinformatics in the last decade or 15 years or so. And really, our guiding questions has been, how much do we understand about the basics of the biology of membrane proteins? And by that I mean, do we understand how they're synthesized? Do we understand how they're inserted into the membrane in the cellular context? What do we know about folding, oligomerization, trafficking, maturation, degradation, everything about the lifespan of a membrane protein, basically. And there are a lot of questions here, and a lot of different groups use a lot of different technologies to address these different issues. And it's really multidisciplinary. So, it's obviously a lot of this is cell biology, but there's also a lot of biophysics, or at least people like me want to reduce it to biophysics as much as possible. Computational studies have become much more powerful these last years, molecular dynamics and coarse graining and so on. And of course, bioinformatics has always been there. So what I'd like to do in the remaining time here is to tell you a little bit about some projects we've been doing more recently that deal with the first two, or possibly a little bit about the third, coming a little bit into the third field here as well, is synthesis and insertion, and maybe a little aspects of folding as well. So the question here is, how do these proteins actually get inserted into the membrane? If you have these very hydrophobic polypeptides, you can imagine that it would not be a good idea if the ribosome would just spit them out into the cytosol. They would presumably aggregate very quickly, and you would have a goo in the cell. So that doesn't sound like a great idea. So as I already showed to you, the way nature has solved this is the way that we could derive from the early Blobel and Sabatini signal hypothesis, is that proteins are inserted, almost all membrane proteins are inserted into a target membrane co-translationally. So they come out of the ribosome and immediately go into the lipid bilayer without passing through a water-soluble state. That's obviously a nice way to solve this. And in fact, Günter Blobel himself provided a an updated model of the signal hypothesis to apply particularly to membrane proteins at some point, where he imagined that the channel through which proteins are secreted through the endoplasmic reticular membrane can also be gated laterally towards the surrounding lipid bilayer, such that when a hydrophobic transmembrane segment gets into the translocon, it doesn't pass all the way through, but it actually goes sideways and becomes integrated in the lipid bilayer. So that was a guiding idea for a long time, and it turned out, again, to be correct. So today we know much more about the structure of these translocon and the structure of the ribosome translocon complex. So here it's actually a composite structure, so this is not, the ribosome is a proper EM structure, and the translocon is a proper X-ray structure, but they've been docked together here just for illustrative purposes. But this docking is fairly well described biochemically and biophysically. So I'm sure it's going to look something like this. So what you have to imagine, or the basic idea, to repeat, how transmembrane segments get inserted into the lipid bilayer, is that you have a signal peptide of some kind early on the protein. This targets the ribosome nascent chain complex to a translocon. So the ribosome docks on top of the translocon, and you now have a conduit that goes all the way from the active site in the ribosome at the end of the tRNA through the tunnel in the large ribosomal subunit and through the translocon itself. So it's a continuous conduit, and if it's a polar piece of polypeptide that's coming through, it's never exposed to the lipid, right? It's just going through this proteinaceous channel all the way to the other side of the membrane. However, if you now imagine that this ribosome, at some point in the synthesis of the protein hits or encounters a hydrophobic segment, a transmembrane segment in the protein, clearly this hydrophobic segment will be polymerized, and it has to make its way down through the tunnel in the ribosome. Doesn't seem to be much of a problem. And as the chain elongates, it's going to go further down, and at some point it's going to be pushed all the way into the translocon channel. And this is where this side, lateral gating happens. So at this point, the system somehow decides that, okay, I'm dealing with a transmembrane segment, should not go all the way through, but instead it should go out laterally and integrate as a transmembrane segment. And this is the process that we've been really interested in, and I'll tell you a little bit more about it. Just to be more complete, this sort of explains how a single spanning protein would be made. But of course, like I showed you, many of these proteins are multispanning, and then you'll have to make the model a little more complex. But there are ways you can imagine that helices sort of go in one by one or maybe in pairs as they come out of the ribosome, and they have alternating orientations relative to the membrane. So there are a lot of questions about how multispanning proteins get inserted, and this is just kind of a conceptual model at this point. And it's definitely not true in detail, but at least it gives you some idea of how to think about the insertion of multispanning proteins as well. So I said there is structural information, and there's actually a number of structures done for the translocon in different states. And for my purposes, this is perhaps the most interesting one. It came out a couple of years ago. And this, because this shows, by sheer luck, these people managed to trap a translocon. This is an archaebacterial translocon with a semi-open lateral gate. So you can see this lateral gate facing you here. So the idea is that it's through this crack in the side of in the wall of the channel that the hydrophobic segments would integrate into the surrounding lipid. There are other structures where this lateral gate is closed, so clearly there is some dynamic going on here. The channel can open and close and gate laterally. So there is a fair amount of sort of basic cell biological and biochemical knowledge about this, including structural information. But what we've, coming from the bioinformatics, I think, and thinking about prediction and algorithms, we always worried about numbers and quantitative information about these things, which cell biologists rarely worry about. So we've been spending some time, probably the last 5 or 6 years, trying to actually get numbers to give more insight into this integration process. basically the idea we had was that if we can come up with hydrophobicity scales based on statistics, maybe we can also derive experimental hydrophobicity scales, if you will, that directly describe and that are measured in the context of this translocon-mediated insertion process. So that's what we were after. It's kind of a biophysical question, but we address it using more cell biological approaches. So just very briefly, the assay that we have been using a lot these last few years, and I should also add we've done a lot of this in collaboration with Steve White, who is at UC Irvine and is a really good biophysicist who knows everything about peptide and protein-lipid interactions, so this has been very useful. But we developed this kind of cell biological assay based on very old existing assays that people in the field have been using for a long time to study protein secretion and also membrane protein assembly. But we wanted to make it quantitative so that we could actually measure insertion efficiencies of different segments as they come through the translocon. So basically the idea is that you make your membrane protein, in our case in a test tube in an in vitro translation system, which is basically just cytosol dumped into a test tube. So all the ribosomes, all the tRNAs, everything you need to make protein are in there. But it's just a bit easier to do it in a test tube than in the cell itself. So you can make protein. You can have whatever protein construct you want encoded on a plasmid, so you can engineer it any way you want. And you then transcribe it into mRNA in vitro, and you add this to a translation system, and that will produce the protein for you. Now, in order to study membrane protein insertion, we need a membrane, obviously, as well in the system. And the membrane we'll be using is what's called rough microsomes. So those are ER-derived membrane vesicles that you can also purify out of cells. But they're basically intact ER membranes, so they have translocons and they have everything in them that's required to reconstitute this process I showed you with the ribosome docking and the translocon and the entry into the membrane. So this is all fine, and this has all been used for a long time, but we wanted to make this quantitative so that we could actually get some numbers. And we did it by a very simple extension of this assay. So what we did from a protein engineering point of view is we put a marker into the protein. And the marker we put into the protein is a short acceptor sequence for something called asparagine-linked glycan addition. So many secreted proteins, almost all of them, and most membrane proteins as well, plasma membrane proteins, are glycoproteins. That means they have sugar moieties attached to them at certain points. And it's a very, the peptide, the recognition sequence you need to get a glycan added is a very simple one. It's asparagine X threonine or asparagine X serine. So that little tripeptide motif, if it's present in the protein, and if that part of the protein is in the lumen of the rough microsomes, that is to say that part has been translocated across the membrane, then it meets an enzyme, oligosaccharide transferase, that can hook on that glycan onto the acceptor sequence. So the glycan gets added to the asparagine. This only works in the lumen of these vesicles. If the acceptor sequence stays on the cytosolic side, there is no enzyme around in the cytosol, so there is no glycan added, despite the fact that you have the proper acceptor sequence. So it's a very nice marker. The addition of a glycan immediately tells you that that part of the protein has made it across the membrane. So that was the key. So basically what we set up was what we're using is a model protein, a host protein, that we've been using in lots of different studies. And when you translate this in this in vitro system, it inserts into the membrane in the way shown here with two transmembrane helices and a big C-terminal domain that's translocated into the lumen of the vesicle. So that's all fine. And the only little extra engineering we do is we introduce first a potential transmembrane segment, this red guy, an H segment, we call them, hydrophobic segment. And we can vary the sequence of this segment at will, so we can make it anything we want very easily. And the whole exercise is to basically make lots and lots of different H segments and measure their insertion, each one of them, and use the data we generate in that way to come up with hydrophobicity values for the different amino acids, natural amino acids that we put into the segment. So a typical H segment, the way we use them, design them, is shown at the bottom here. It's just a stretch of alanines and leucines. It's 19 residues long, and that's the right length to span a membrane. And of course, we can just fine-tune the hydrophobicity of this segment by increasing or decreasing the number of leucines. More leucines, more hydrophobic, more alanines, less hydrophobic. So it's very easy to titrate the hydrophobicity of these segments. Now, how do we know if a given H segment gets inserted into the membrane as a transmembrane segment, or if it's just translocated across the membrane? Well, that's when these glycosylation sites come into play. So we engineer one site on each side of this H segment, so two acceptor sites. And the whole idea, I'm sure you've already realized, is that if we have an H segment that has the right properties to be transmembrane, we get the conformation shown on the left, where only the first site, the G1 site, has made it across the membrane and receives a glycan. On the other hand, if the segment doesn't have the right properties to engage in the membrane, but just translocate across, both of the sites will make it into the lumen. So the protein will receive two glycans, one on each site. And it's very easy experimentally. You just run the protein out on a gel, and you look at the size of the protein, because the glycan addition is big enough that you can see a clear size shift depending on how many glycans have been added. So here is an example. Here is a given sequence at the bottom, 4 leucines, a cysteine in the middle in this case. And you see that when we translate the protein first without microsomes, minus RM, of course we don't get any glycan added because there is no membrane, there is no oligosaccharide transferase. But if we have the membrane present during translation of the protein, you see that most of the protein shifts upwards in molecular weight. So it's a little bit heavier than the original naked protein. And that's because one glycan has been added in this case. You see the weak upper band, that's what you get when 2 glycans are added. So this particular segment with 4 leucines and a cysteine, and the rest is alanine, is a fairly good transmembrane segment. It inserts to, say, 90% into the membrane. 10% slips through, but most of it becomes transmembrane. So it's a pretty good transmembrane helix. If you mutate the cysteine in this particular case to a proline, so that's the only change we do, now we get the result shown at the bottom, where now almost everything receives 2 glycans. And that is because the whole H segment, the whole C-terminal domain, now makes it across the membrane into the lumen. So you see, so that single mutation from proline to cysteine in this particular background is enough to shift from something that inserts well to something that doesn't insert. So it's a fairly sensitive assay. And I think you can imagine that, it's now just a question of, well, first quantitating these bands, of course, by just measuring densities. We can define something like an equilibrium constant between the inserted and non-inserted forms by just taking the quotient between the 2 bands. And we can convert that into something like a free energy difference between the inserted and non-inserted states by just minus RT log K. So we can get numbers. For each particular segment, we can measure the free energy of insertion in this way. And now it's just a question of making hundreds of different segments to tease out how each individual amino acid affects the overall insertion probability or the insertion free energy. And we can do it as a function of position along the H segment as well. So we can put the particular amino acid we're interested in at the N terminus or in the middle or at the C terminus of the segment, and we can check whether it affects insertion differently depending on position. So clearly, the next thing you do is you do these scans for all the 20 amino acids, and you try to tease out, how they all individually contribute to membrane insertion. When you do that, you get basically the blue curves here. So those are the 20 amino acids. The blue curves show you how, say, an aspartic acid, a D in the top row, how that affects the overall insertion free energy as a function of position in the segment. So the aspartate, you see, has, quite a... It's very much dependent on position in the case of aspartate, the D residue. So if you put the D in the middle of the segment, it's very costly. It increases the free energy of insertion by 2 kcals per mole. So it reduces the insertion frequency, right? If you put the aspartate near the ends, it's not nearly as bad. It's still positive, but it's only, say, half a kcal per mole. So aspartic acid really depends on where you put it, how much the cost is. If you take a hydrophobic residue like leucine, L on the second row, you see the blue curve is always underneath the 0 point, so leucine always increases insertion. It has a negative free energy of insertion. And it also doesn't matter much where you put it. It's pretty independent of position. And that's the general rule, is that charged and polar residues, of course, reduce insertion, they increase the free energy of insertion, and they depend quite a lot on position. So the more central it is, the worse it is. Whereas the hydrophobic residues, of course, have negative values, increase insertion, and actually don't depend very much on position, none of them. So that's a fairly simple way to summarize these. In red, just as a comparison, those are statistical free energies defined or calculated, computed from known X-ray structures by just calculating the frequency of incidence of each amino acid as a function of depth in the membrane in the known X-ray structures, and converting that into a statistical free energy minus RT log probability. And it's kind of interesting to see that what you see in the real structures in terms of where you find different amino acids actually is not too far away from, and in some cases actually too close to be believable to what we see with this very simple insertion assay, right? So for aspartate, the two curves superimpose basically. And of course they're defined in completely different ways. And in some cases there is more of a discrepancy, but then there are some reasons why that happens. But so by and large, we have a pretty good idea of how different amino acids impact insertion now in the context, in this fully biological context. Just as a fun extension of this, recently we teamed up with a group in Japan, Hiroaki Suga's group in Tokyo, to expand beyond the 20 natural amino acids to non-natural amino acids, where we actually have full control over the side chain chemistry. So we can put any side chain we want now, at least into one position in these segments, still maintaining the full biological system. And this is done, I won't go into details, it's done by putting a stop codon in the position where you want your non-natural amino acid. You then add to your reaction translation mix a so-called suppressor tRNA. It's a special tRNA that can read stop codons. And before you do that, you add your non-natural amino acid. You couple that to that suppressor tRNA chemically. So you have a chemically prepared suppressor tRNA with a non-natural amino acid. You put that in the translation mix. When the ribosome gets to the stop codon, it then introduces that non-natural amino acid, and then it continues. So then we can repeat the same experiments, but now with full control of the side chain chemistry. And actually, this is just the way we add these non-natural amino acids, which I won't go into. But just to show you what you can learn from that, the first thing we did was we just made side chains with increasingly long alkyl side chains, just adding one methyl group, two methyl group, three methyl groups, just a long linear string of methyl groups or carbon atoms up to 8 long. That's the longest we tested, so from 1 to 8. And we just checked that how does a single, addition of a single methyl group affect the insertion probability or the insertion free energy. And if you focus on the red dots, that's the result we got. So that's when we put increasingly long side chains right in the middle of our transmembrane segment. So we have the accessible surface area that, of course, increases as we lengthen this one amino acid. And on the y-axis, you have the change in free energy of insertion measured by this glycosylation assay as we increase the length of the side chain. And you see the red dots, so those are just adding one methyl after the other, fall on a very nice straight line. And the slope of that line actually tells us that for each square angstrom of non-polar surface area that we add to the hydrophobic helix, we gain about 10 calories per mole of insertion free energy. So that's kind of, if you want, that's the number for the hydrophobic effect in this, biological insertion system. basically. And one can then compare this, and this number actually has led to a lot of work in molecular dynamics and biophysical chemistry, trying to see what match this up, because it's not the same number that you get if you use a pure biophysical system, say buffer and lipid, and you measure partitioning of side chains or simple hydrocarbons. You get about twice that number. So this has really spawned a lot of work now, trying to reconcile these two, trying to figure out why they're different. But of course, from all these numbers, coming back to prediction, one can also try to predict, of course, that the free energy of insertion now for any particular segment. And we do that. It turns out we can do that pretty well by just summing up the individual values for the individual residues that that are included in any given segment. Take the numbers from these profiles that I showed you, just sum them up, and we also measured using other constructs how the overall length of the segment impacts the free energy of insertion, and that's the second order polynomial on the right there. So that's also been those are also measured numbers to allow us to take different lengths of hydrophobic segments into account. So all of these numbers are just measured numbers that you see there. And this does a decent job of predicting for any given segment its free energy of insertion. And generally we get the right number within, say, half a kcal per mole or so when we do this. So we figured, coming back to topology prediction, which of course we and others have done using, sophisticated statistical models, HMMs and what have you, we said, what about using these numbers instead? We've measured this now. We don't have to extract it by sophisticated statistics on known sequences and structures. So we made a very simple predictor, topology predictor, where we used these measured numbers and only had two adjustable parameters beyond those measured numbers. And actually came up with a predictor that it doesn't do better than the best HMMs or the best machine learning methods, but it basically matches them. So from a kind of physicist perspective, I like this because almost everything is now based on measured parameters and not sort of black box training things, and we get performances that are pretty good, more or less what you get with the best other methods. Another thing you can do when you can calculate or predict these free energies of insertion is, of course, you can look at real proteins and see, the known transmembrane helices from structures, how do they score on this scale? So what I show here is a couple of different types of proteins. Single-span proteins on the left, so those are the transmembrane helices from single-span proteins. This is the free energy of the distribution of the free energy of predicted free energy of insertion, and you see they all come out as negative, which is nice because after all we know that they are transmembrane, so they should come out as negative. And on the right you see secreted proteins. So we've just gone through about 1,000 secreted proteins, and we've looked for the most hydrophobic segment in each one. And you see the distribution of those numbers on the right, and especially interesting is that all of them come out positive. And of course, secreted proteins have all gone through the translocon and none of them got stuck, right? So there shouldn't be any segments with a negative delta G value. And we don't find any, basically. And the overlap is one sequence, I think, between these two distributions. So it's pretty nice. But what's really interesting is when we do this exercise for multispanning proteins, so we take the helices from the X-ray structures, the stuff that's in the membrane, and we do this, and then we get the middle distribution, which is, overall hydrophobic. It's on the negative side, but there is actually quite a big tail of it that's positive. So there is a good number, 25-30% of all the helices in the known structures come out positive on this scale. So the question is how can that be? How does nature trick the system, basically? And I'll just show you as the last slide here, or the last little piece is. This is something we're doing right now, is we're trying to figure out the tricks. And we've found that there are a number of tricks that nature is using to get around this problem. Because for functional reasons, it's often required to have a charge residue or something in one of these transmembrane helices, so it shifts it to the right. I'll just show you one which is quite simple, but still quite interesting in its implications. this is a case we found. It was a postdoc, Annie Kauko, in Arne Elofsson's group, who just went about essentially coloring the known structures, the helices and known structures, using the predicted free energy of insertion. And she found this one example, which is trimeric structure, as you see on the top. It's a transporter. But when she looked at the monomers, and the monomer, and that's on the bottom here, she found that quite a few of the transmembrane helices actually had pretty high delta G values. So you see their color there, and they're clearly positive, quite a few of them. But then she did an interesting extra bit. So she said, well, what if we look not just in the membrane itself, but if we also look a little bit outside the membrane? Maybe there are more hydrophobic pieces out just near outside the membrane. And she found a couple of good examples. So, for instance, this helix here, which is clearly transmembrane in the protein, but it has a very funny shape. It's broken, and it's not very hydrophobic. But when she looked nearby, she found this very hydrophobic helix, which is just an extension to this transmembrane helix, but it sticks out into the cytoplasm. And it's actually forming the trimeric interface, so it's holding the trimer together. But it's very hydrophobic. So what we could show, and I'm not going to go into how we did it, we used, again, glycosylation and mapping and so on, but what we could show was that, in fact, when the monomer inserts in the membrane, it's the dark blue piece that's the transmembrane segment. And this funny-looking thing on the left is actually outside the membrane in the monomer. So presumably during the trimer formation, during oligomerization, you get a rearrangement, quite a dramatic one here, which pushes the blue one out and brings in this other part of the protein into the membrane. So here is clearly a case where it's a folding, sub-insertion or post-insertion folding event that allows this particular protein to have some very hydrophilic helices and even some strange-looking helices spanning the membrane, where they have some important functional roles, right? So this, I just want to show you this one case because, well, it's an easy one, and also I think it has some interesting implications for 3D structure prediction, because this might not be so easy to predict, actually. So don't try this protein in your... Okay. Right, so this is, trying to summarize 30 years of, or a few chosen examples from 30 years of membrane protein work. And I wish I had a really nice, summary take-home thing to tell you that would, forever put membrane proteins at the top of your head or top of your mind. But the best I could come up with thinking back is that why work on membrane proteins? Well, it's simply a rich source of very fun science, I think. At least it has been for us, and it continues to be for us. A really nice area where, you can take bits and pieces from so many different techniques, from so many different approaches, from wet lab, from biochemistry, from cell biology, biophysics, computational biology, bioinformatics, and it's all relevant. And it all helps paint the picture of what these interesting molecules look like and what they do. So with that, I'll just give the ordinary thanks, of course, to funding agencies, but as always, it's the people that matter. And very fortunately, about a year ago, I was able to collect almost everyone who'd ever been through the lab for a celebration. So these are all the people that have done the work. Thank you so much.
Thank you, Gunnar. Questions? Do I have some light in the room?
Oh, yeah, there's one.
Let's see here. I don't hear any sound. There we go.
Yeah.
I was interested in your measurement of the different amino acids and how they translocate or not across the membrane. It's a new way to measure their hydrophobicity and hydrophilicity. So one thing that struck me was cysteine was exactly flat.
Yeah. Okay. So yes. So histidine is
Cysteine.
it's kind of in a way it's kind of an artifact. So what histidine?
I was talking about cysteine.
Cysteine. Okay. Cysteine. Yeah. Cysteine is pretty flat. It's it's as hydrophobic as, I don't know, serine or something. It appears. But of course, it's a single one. It doesn't make disulfide bonds in this context. So it's a fairly, sort of mildly hydrophobic residue on this scale.
And we know that there are those other two unusual but natural amino acids, selenocysteine and pyrrolysine. I don't know if they play a role in membrane proteins or not.
I mean, selenocysteine, I think, so far only plays a role in the membrane protein world when you do crystallography. So, and we haven't actually measured it, but I don't suppose it's very different from normal cysteine. Pyrrolysine, I have no idea, but again, I don't think it's something you find a lot of in membrane proteins.
So, here.
Yeah.
Really beautiful talk. I would like to ask you, in the case of ion channels, such as those that need to really to be sensitive to voltage and so on, they do have a helix which is extremely hydrophilic.
Yeah.
And what's the trick of it in this case?
Right. So what you're talking about here is something called the voltage sensor domain, which you find in many ion channels, that reacts to membrane voltage by shifting its position in the membrane.
Yes.
And it's particular, there's a so-called S4 helix, which has every third residue is an arginine or possibly a lysine. Mm-hmm. So if you go across the helix, everything else is very hydrophobic, but you have these regularly spaced arginines or lysines. So obviously this is one thing we measured. And it turns out that if you take those guys, they certainly don't insert efficiently, but most of them insert measurably, I would say. So say 30%, 40%. I think we had one that's even inserts to 50%. And the neat thing is that we can explain those, or we can predict those insertion efficiencies roughly, simply because arginine has this strong dependence on position. So you have 4 arginines spaced along this helix, but it's only really one of them that's kind of central in the membrane. The others are by necessity sort of towards the ends, right? So that helps a lot. But it doesn't explain the whole thing. And the rest of it, we think, is mostly helix-helix interactions. So some of these charges gets buried by counter charges on neighboring helices.
I see.
So that's probably how it really works.
Thank you.
Yeah.
Yes, on your right. I was wondering how this translates into these channels that transport DNA and nowadays are being used to create new sequencer technologies. And there is obviously the need to engineer this, to slow down the translocation. Yeah. And I was wondering if you have any insights on how this works and how this work you do can help those.
It's not something I've thought about. I think these channels they use are either porins or possibly some sort of viral channels, or I'm not exactly sure what they are. But I mean, it's relevant insofar as you would need to have whatever channel you make. If you want to put it in a standard lipid bilayer, at least, you would have to have a surface that's sufficiently hydrophobic to keep the thing in there, and that you could probably estimate from these sort of numbers. I think I really don't know how they have been designed, but I'm sure they're taken out of some membrane and put into a simplified membrane for this work. But I really don't have any good comments on it.
Right here in the middle.
Straight
ahead. So it's a little bit of a naive question, but I'm curious as to what the diversity of these sort of adapter machineries is, and how do they find their specificity? I mean, if different classes of proteins are being synthesized, is there some kind of, I mean, is it possible that perhaps 5' UTR, UORFs are playing some role in sort of recruiting these machines, and what is their evolution?
What is known is that different membrane systems have different machines. So the one I showed you is the one in the endoplasmic reticulum or in the inner membrane of bacteria. They're very similar. But you have a very different type of machinery in the inner membrane of mitochondria, for instance. So for import, or outer and inner membrane. Wow. So for import into mitochondria and for assembly of membrane proteins in mitochondria, you have a different type system. We've tried to measure hydrophobicities in that context as well, and we find fairly similar numbers. So from that point of view, they seem to behave quite similarly, but the actual architectures is very different, and the basic mode of function is quite different.
When you say architecture, do you mean that it's convergent evolution? They don't have a common.
Yeah,
wow.
they don't have any, sequence similarity or even structure similarity. So those are different. They have a different evolutionary background.
And what's the diversity overall? I mean, how many of these are known?
Well, you have specialized systems in the outer membrane of bacteria, for instance, and there's probably, let's say, 10 different types or something. It's not hundreds of different types. Yeah.
Thank you very, very much.
How do the membrane proteins degrade?
So there is this whole field in cell biology about protein degradation, especially in the endoplasmic reticulum, misfolded proteins that are prevented from escaping or getting out of the endoplasmic reticulum. And basically they seem to be degraded by similar mechanisms, whether they're soluble proteins or membrane proteins. They're basically pulled out through some kind of, at this point, ill-defined channel in the endoplasmic reticulum membrane. They're pulled back out into the cytosol. They're ubiquinated, and then the proteasome degrades them. So there is some kind of ATP-driven translocator that can pull membrane proteins out of the membrane, back out of the membrane, put ubiquitin on them and target them for degradation. So that's one important degradation pathway, at least.
Thank you.
Last question.
Yeah, thank you for the talk. So are there certain differences in half inner cell part and a half outer cell part?
Sorry, in the...
So the inner membrane proteins, half of the inner cell part and the outer cell part. So do they have certain differences?
Yeah, I mean, so the main difference between let's say the extracellular parts and the intracellular parts of especially multispanning proteins, the most obvious difference, at least, is this positive charge bias. That you have many more positive charges on the inside than on the outside. But of course, there are also finer differences, and for instance, disulfide bonds you normally don't find on the inside, but you find them on the outside. You have these glycan additions that I talked about. The glycans are always on the outside, never on the inside. And there are other modifications that only occur on one side and not on the other side. So yes, they are in different environments, and they're somewhat adapted to. The two sides are adapted to different environments, basically, and there are a number of characteristics that reflect that.
What about loops? I mean, if you want to do kind of your protein loop structure prediction, so on outer part and an inner part, do you think?
Okay, so you want to predict loop structure. I don't know how that would translate in this case, if you would need, sort of special predictors for the outside and the inside. This is something for Burkhardt. I don't know.
Thank you again, Gunnar. We have to unfortunately move on. We did that already. We can't get enough of it. Thank you.